Welcome to our channel Bio by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, we have learned about glycolysis and Krebs cycle under the heading Respiration in higher plants. Now, as we understood that during glycolysis and Krebs cycle, what NADH2 and FADH2 are produced, they supposed to undergo oxidation, breakdown, as stored energy to be released as a byproduct of respiration process. So, where do that NADH2 and FADH2 get completely broken, oxidized in mitochondria? So, they have to transfer that NADH2 produced during glycolysis must be transferred to the mitochondria, as well as FADH2 are already and other NADH2 produced during Krebs cycle are already inside the mitochondria. But where this process occur, how this NADH2 and FADH2 undergo oxidation as a byproduct of that proton and electron produced. So proton are supposed to produce the ATP, but electron release must be transported through oxidizing reducing substance present in the inner membrane. Therefore, let us understand what is electron transport system. Electron transport system. Very important part of process of respiration, as these enable that process to release the energy stored in NADH2 and FADH2. As we know, structure of mitochondria. Mitochondria has inner wall having finger-like projections called crista. These finger-like projections are infolding of inner wall, and ATP synthetase enzymes present in mitochondria directing towards inside. That F1, F0 directing towards inside. These are the ATP synthetase. That F1, F0 is present inside the membrane, F1 is locating outside. This is the outer membrane of mitochondria. This is inner membrane of mitochondria. We can expand this inner membrane of mitochondria as electron transport system. That is, electron transport occur through inner membrane of the mitochondria. Now, when we do, as like in mobile, we expand like this. Suppose this is expansion of inner membrane. So, as to understand how the best understanding how these 5 complex 1, 2, 3, 4 and 5 are located in inner membrane to receive and pump H positive ion outside the inner membrane, as well as electron are transported across the membrane. Here complex 1, I do write here, complex 1 is NADH2 dehydrogenase, that what enable NADH2 nicotinamide adenosine dihydrogen phosphate to oxidize and release that 2 molecule of NADH2, release that proton, that 4H, which are directly transported across the membrane, as well as that electron released, that proton release combined with FMN forming FMNH2, and finally this proton combined with ubiquinone forming UQH2, and this what UQH2 further release as along with this UQH2 that hydrogen with the oxidation of FADH2. So there FADH2 oxidized into FAD, releasing 2 more proton, transferred to ubiquinone to UQS2, this 2 more proton along with this, with the oxidation of FADS2 combine with UQ to form UQH2. So total 4 H positive ion are transported here. But as this UQ combine with UQH2 further release electron, this released electron are transported to cytochrome B to sulfur to iron, and iron to cytochrome C. Now this electron which transported from this UQS2 one proton is released outside. Along with this proton from pigment cyst, this complex 1. Here this is a complex 2. That what FADS2 oxidized to form FAD, proton are released combined with ubiquinone to form UQS2. An electron release is transported over cytochrome B. To understand this, we have this diagram. Here you can see with the oxidation of FADS2 that proton release combined with ubiquinone. Now ubiquinone release that proton as well as electron transfer to cytochrome B to sulfur to iron, and iron to cytochrome C1. So here you can see, here it is written cytochrome C1. Now further this electron from cytochrome C1 is transferred to cytochrome C. So here cytochrome C to cytochrome C to cytochrome A to A3. This is complex 4. This is complex 4. This was complex 2 and this was complex, suppose this was complex 3. So here when electron reach to cytochrome A to A3, then it is released inside. These 2 electrons are utilized to ionize oxygen and combine with that proton when they are transported through. This is the inner membrane where this is F0 unit, this is F1, it is a catalytical molecule F1 where this proton now from this cytochrome A3, here ubiquinone again ready to receive 2 more proton and from UQH2 and release this total 4 hydrogen because 4 protons are transferred through. Now these protons which are transported, now they are transferred from its higher concentration to lower concentration, that is from intermembrane space to matrix. When these protons are transported, every time 2H combine with this oxygen forming water molecule. So finally this ionized oxygen is used to control the proton and form the water. This formation of water is the final stage of that oxygen we inhale enter into the mitochondria. Now as a part of just revision what I did explain to you, here there are total 5 complex. So role of 5 complex to be understood in a sequence. So see, complex 1 is the process of oxidation of NADS2 to NAD, leaving 4 hydrogen directly into the intermembrane space, as well as that electron release accepted by flavine mononucleotide with proton. That proton from flavine nucleotide are transferred to ubiquinone to naturally outside, where that complex 2 also at the same time oxidize this FADS2 into FAD, releasing proton combined with UBQS2 also release along with that proton release from complex 2, complex 3 together into the intermembrane space. Further electron is transferred from cytochrome B to sulfur to iron, iron to C1 and then C1 to C. So that electron from complex 2 is directly transferred to complex 4 through C to C1 and C1 to C and cytochrome C to A, 1 to A3. So finally electron released into the matrix. This is the inner membrane space through which that electron is received and transported across, so that finally reach to the matrix generating H2 molecule by controlling H positive ion. This proton get accumulated into this inner membrane space. This is intermembrane space where protons are accumulated developing potential energy so as to transfer them from higher concentration to lower concentration, generating ATP molecule by the combination of ADP plus inorganic phosphate. So finally ATPs are produced over here. So electron transport system is the process of stepwise oxidation of NADS2 and FADS2 so as to release the proton into inner membrane space as well as electron through the inner membrane through electron transport chain as to release into the matrix, and proton which are accumulated outside the inner membrane create higher potential in between that outer and inner membrane so as to transfer from its higher concentration to lower concentration. Leaving potential energy is used to combine this adenosine diphosphate with inorganic phosphate to generate the ATP. So finally ATP are produced. So see, these five systems together, five complex together work to have the production of ATP inside the mitochondria. So finally this ATP are produced in matrix and remain stored, and by the time as and when they are required they can be used as a energy currency. Therefore mitochondria are known as powerhouse of cell. So see, as I did explain to you, here it is clearly mentioned how that protons are pumped outside the inner membrane by these oxidizing reducing substances by gaining proton and electron. Subsequently, while gaining proton they also gain electron, while leaving proton they left electron. So electron finally transferred through this electron transport system into the matrix to ionize the oxygen and combine with proton to form the water. So water produced during respiration, that is C6H12O6 plus 6O2 lead to produce 6 CO2 and 6 H2O. That CO2 what we understood, that was released during Krebs cycle decarboxylation process. Here the water is produced, so 6 H2O are produced during that respiration as a final product inside the mitochondria, that is called metabolic water. So when we inhale the oxygen, it gets into the mitochondria and there it is used, that is the final destiny of the oxygen, to use to control the proton. If we do not take the oxygen we feel suffocate. It is because of accumulation of proton into the mitochondria, because increase in H positive ion concentration cause the acidity, and acidic condition is dangerous for organelles as well as cell, which enable us to feel the suffocate. So for the time when we do not inhale for 1 to 2 minutes we feel very suffocated, because by the time hydrogen ion concentration increase in mitochondria and thereby there may be death of the mitochondria. And if we do feel hypoxia, that is very less oxygen, we may get into the low energy level and thereby may have very tense condition to the brain and body. Therefore we continuously need to inhale the oxygen so as to control the H positive ion generated during this electron transport system for the production of energy. I hope you got it, what exactly the role of electron transport system and mitochondria in the production of energy. As it is mentioned here, succinic dehydrogenase. Yes, here this FADS2 oxidize into FAD in presence of succinic dehydrogenase. So this succinic dehydrogenase can be called as a complex 2, and that is what we have to remember as a complex 2 here, succinic dehydrogenase, as it is being used to hydrolyze FAD into FADS2 to FAD. By the time that hydrolyze into FAD and then succinate get converted into fumarate. So succinate dehydrogenase with the oxygen of NADS2 produce succinate and fumarate for releasing of proton. So here this is electron transport system which is given in your book. Same I did explain over here to have the understanding how that energy is being produced during electron transport system, and byproduct is water. So here it is clear to you that where the electron transport is located, system is located in the inner membranes of the mitochondria. Inner membrane of the mitochondria consists of the 5 complex, complex 1, complex 2, complex 3, complex 4, complex 5. So these all the complexes, they are involved in the electron transport system, as well as this complex 5 is ATP synthetase which present in the inner membrane directing towards inside so as to produce and accumulate ATP inside the mitochondrial matrix. That is what I did explain here. So practice this diagram so as to have accuracy to understand how electron is transported Once again, electron is transported from flavin mononucleotide to ubiquinone, cytochrome B to S, iron cytochrome C1 to C, C to cytochrome A1, A3 to matrix, and that is what question can be asked in NEET. Electron is transported through, so we have to remember this cytochrome B, cytochrome C1, cytochrome C1, cytochrome C, cytochrome C2, cytochrome A1, A3, and finally in matrix. You also have to remember complex 1 as a NADH2 dehydrogenase. Complex 1 is NADH2 dehydrogenase, is a complex 1, and this ubiquinone UQS2 is a complex 3. Then succinic dehydrogenase is a complex 2, and this cytochrome C1 transfer that electron through cytochrome C into A1, A3, this is complex 4, and this ATP synthetase, this enzyme is located here as a F0 F unit. This is a ATP synthetase. ATP synthetase is a complex 5. So I hope you did understand what is electron transport system is a little bit complex, but once you practice this one and enable to draw accurately, then it becomes very easy to explain. After all, this system is nothing but oxidizing. It is a complex system made up of 5 complex of oxidizing reducing substances to receive and release electrons so as to transfer through. Therefore, this electron transport system can be called as oxidizing reducing substance, finally to release the energy stored in NADPS2 and FADS2 in the form of ATP. I hope you did understand. You just go through, see this video again and again to have the clear understanding, and even if you are in doubt, you can ask in comment section. I appeal you to like our channel as well as subscribe it and press bell icon so as to receive videos time to time when they are uploaded. Thank you.